presentation, we'll conduct a comparative look at hexokinase and glucokinase. Hexokinases are a family of enzymes or isozymes. Isozymes are homologous enzymes within a single organism which catalyze the same reaction but differ slightly in structure, catalytic properties, and regulatory properties. Basically, isozymes provide different avenues of regulation for the same reaction at distinct locations or time in order to meet the specific physiological needs in the specific tissues. Hexokinases phosphorylate using ATP a hexose, or a 6-carbon sugar, to a hexose phosphate, a 6-carbon sugar with a phosphate group attached. Looking at this picture, hexokinases react with the CH2OH group. As long as the 6-carbon sugar has this moiety, a hexokinase will phosphorylate it. Hexokinase 1, which I will just call hexokinase, hexokinase 4, which I will call glucokinase, both react with glucose, but we will see some differences. These are the structures of hexokinase and glucokinase. Basically, just note that the structures of these enzymes are a little bit different and a little bit similar, which is part of the reason they regulate glucose differently. Another note is each enzyme has 2 lobes, one lobe here and here, one lobe here, one lobe here. One lobe binds with ATP and the other with glucose, and upon the conformational change, these 2 substrates will be located in a single catalytic center, which will allow for the phosphate group transfer. Let's start by looking at some of the main differences between hexokinase and glucokinase. Hexokinase is located in all tissues, has a low Km, uses feedback inhibition, and is a regulatory step in glycolysis. Glucokinase is located in low concentrations in many tissues, but concentrated in the pancreas, which I will only discuss briefly, and the liver, making it tissue-specific. Glucokinase also has a high Km, which doesn't use feedback inhibition and is used to help regulate blood glucose levels. A Km value is the substrate value at which the reaction rate is half Vmax. As I won't be getting into reaction kinetics, what is important to note here is a low Km value means the enzyme is active at low concentrations of substrate. A high Km value indicates the enzyme is only active at high concentrations of substrate. For the means of this presentation, the substrate of hexokinase and glucokinase is glucose. Feedback inhibition means the enzyme is sensitive to the level of product being formed. The product for these 2 enzymes is glucose 6-phosphate. Therefore, hexokinase activity will be slowed when the concentration of glucose 6-phosphate reaches a threshold value. Glucokinase will be active regardless of the concentration of glucose 6-phosphate. But remember, glucokinase will only become active at high concentrations of glucose. When the glucose concentration goes below a threshold, glucokinase activity will be slowed. Hexokinase is a regulatory step in glycolysis. Glucose can permeate the cell membrane easily. When hexokinase phosphorylates glucose, glucose becomes stuck inside of the cell. This activity helps regulate glycolysis within a cell. Glucokinase is liver specific and helps to maintain blood glucose levels within the body. This will be discussed a little later. Glycolysis is a sequence of reactions that metabolizes 1 molecule of glucose to 2 molecules of pyruvate with the net yield of 2 molecules of ATP. The first step of glycolysis is seen in this reaction below. We get glucose. We are going to make glucose 6-phosphate using hexokinase, and an ATP will be converted to an ADP. This is the first regulatory step of glycolysis and serves 2 functions. 1, it prevents glucose from leaving the cell, and 2, the addition of the phosphoryl group destabilizes glucose, facilitating further metabolism in the glycolytic pathway. This enzyme requires an Mg2+ ion, forming a complex with ATP to mediate the transfer of the phosphoryl group. When hexokinase binds with glucose, a large conformational change occurs. The 2 lobes that I pointed out earlier of the enzyme structure move toward each other, trapping the glucose for the phosphate group transfer. So what does all of this mean in terms of differences between hexokinase and glucokinase? Hexokinase is located in all tissues because all tissues need energy. Glycolysis is the first pathway in cellular respiration where most of our energy comes from. By having less specificity, this enzyme can start glycolysis in all tissues. With a low Km, hexokinase is active at low glucose concentration. When there isn't much glucose in the body, but the tissues still need energy, this enzyme is still active and able to start the tissues on the pathway to cellular respiration in order to provide the necessary energy. Hexokinase is also inhibited by its product, glucose 6-phosphate. When large amounts of glucose 6-phosphate are detected, The cell is told that it no longer needs glucose for the energy, and the enzyme slows its activity. This prevents hexokinase from using up all the glucose, which needs to be used for other functions in the body. The liver helps limit the amount of glucose in the blood when there are large quantities of glucose available. The liver does this by storing the glucose as glycogen, which can be broken back down into glucose when the blood glucose levels drop. The liver has high concentrations of glucokinase, which specifically targets glucose over other 6-carbon sugars. Glucokinase converts glucose into glucose 6-phosphate when glucose enters the liver cells. This traps glucose into the liver cells, removing glucose from the bloodstream because during a fed state, there is excess glucose present. If we take this back a few steps, insulin is released from the pancreas during a fed state, which stimulates the storage of fuels and the synthesis of proteins. Insulin stimulates this glycogen synthesis and glycolysis in the liver. In other words, insulin released after a large meal stimulates the glucokinase to trap glucose in the liver for storage, so that during a fasting state, the storage can be broken back down into glucose and used to make energy in other parts of the body. This is a diagram of glycogen, which is broken down starting at the ends on the outside during times when glucose is scarce in the body. Glucokinase is a regulatory step to trap and destabilize glucose for further metabolism. This reaction is the same as hexokinase. It uses ATP and the Mg2+. Glucokinase also experiences an induced structural change upon the binding of glucose. So what makes glucokinase different? Recall that glucokinase has a large Km. This means glucokinase is only active when glucose concentrations are high and in excess. Thus, if glucose in the bloodstream is limited, glucokinase allows other tissues like the muscle and brain, which need a lot of energy, first dibs on the glucose rather than using all of the glucose for storage. Also, glucokinase is not inhibited by high concentrations of glucose 6-phosphate. This means glucokinase continues to trap glucose in the liver cells until the concentration of glucose drops below the threshold concentration. This allows the liver to store as much glucose as possible while it is available, ensuring the body will have enough glucose storage during times of fasting. We also know glucose is liver specific. The liver is one of the main monitors for blood glucose concentration. This is because all of the excess glucose is stored here. In other words, this isozyme evolved in the liver to provide the storage of the glucose for later use. This is in contrast to hexokinase, which traps glucose for energy use in other tissues. Basically, these enzymes use the same substrate, converting it to the same product. Both enzymes experience conformational changes, which is due to the similar structure. Hexokinase regulates glycolysis for the energy use in all tissues. This isozyme is active at low glucose concentrations and is blocked by the product, signaling enough energy has been made. Glucokinase stores excess glucose for later use in the liver and is partly activated by the pancreas. This isozyme is active at high glucose concentrations and is not blocked by the product, allowing a lot of glucose to be trapped in specific cells, which create the glucose storage molecule glycogen. If you would like to know more about glucokinase and hexokinase, these are some good places to start.